All right, welcome to Chapter 8. Chapter 8 is all about mutation and mutant selection. So in microbiology, we are always looking at how cells mutate naturally as well as how they mutate under certain conditions because we want to mutate cells so that they can do different things that they normally couldn't do. And then if we are going to mutate a cell, make a cell unique, have characteristics, we have to be able to select for those organisms so that we can find them and grow them. And so let's think about what a mutation is initially. So mutations are changes on the genetic level, and mutations aren't good or bad. They just are changes. If a mutation allows an organism to survive better, be better adapted to the environment, then that mutation will stick around and become more common. But in general, a mutation is not considered a good or a bad thing. So down here, this is a figure I got off of Google Images. Here we have a single cell that is just a normal bacterial cell. Oh, hello. Normal bacterial cell. It does not have any bacteria or antibiotic resistance. It grows and grows and grows, and eventually there are billions of identical cells with a few outliers that have mutated. Because anytime you have billions of replications, there's likely going to be at least one or two cells that are mutated. So if the environment changes, and in this case some drug is put into the environment, it kills off all those non-resistant cells, but those drug-resistant cells survive, and then they start replicating and producing identical copies and clones of themselves. And now you have a colony of billions of drug-resistant microbes. That's how mutations and evolution occurs. All right. So you can see mutations occurring both vertically and horizontally. So here we have a parent cell that is going to replicate and copy its DNA and produce two daughter cells. And this daughter cell, a mutation has occurred. And so this is just spontaneously, some change in the genes. And so now when this cell replicates and produces daughter cells, the daughter cells are going to have that same mutation. In horizontal gene transfer, you have a parent cell, and you have two parent cells in the same generation that transmit genes either to each other or from one cell to the other cell. Sometimes they can swap, otherwise one cell will take from the other. So there are spontaneous mutations, and then there are induced mutations. Induced mutations are mutations that occur because of chemicals or radiation, whereas spontaneous mutations are ones that just occur randomly. They're very uncommon. So the first one we'll talk about is a base substitution mutation. This is called a point mutation, where one nucleotide changes. And so here we have DNA, that's the parent DNA. It's split, and you have the replication of each side. Here's the TTC, and here's the AAG. And instead of AAG forming, you have AGG. And so this actually will cause a mutation as the new daughter cells, as this daughter cell replicates. So now you have AGG TCC versus the wild type AAG TTC. And so base substitutions can cause a silent mutation where you produce the exact same amino acid. Since we have multiple codes that code for the same amino acid, this is actually very common to have the silent mutation, and it decreases mutation rates. A missense mutation is where you have, instead of the wild amino acid, you now produce a new amino acid in place. That can cause the protein to be differently functional, or it could be potentially non-functional. And then the last is called a nonsense mutation, where instead of producing the amino acid you're supposed to, you produce a stop code. And so this oftentimes causes the protein to not form at all, or the protein to be non-functional. Silent mutations, or not silent mutations, I'm sorry. Nonsense mutations are often called knockout mutations because they inactivate genes, they stop genes from working, and they can actually cause death of the cell. Silent mutations don't cause any problems with the cell because the protein will be the exact same. And then missense produces those differently functional proteins. I'm not going to play this, but this is something you can look up if you want to. It's just mutation by base substitution. Look it up on Google, okay? You can also have a deletion or an insertion of nucleotides, and this leads to a frameshift mutation. So the frame of the reading that you have changes. So think about it. We read our DNA 3 nucleotides at a time. And so if you have a nucleotide that leaves or a nucleotide that moves in, it changes how you read the DNA. And so now let's look at that. The cat ate the rat. Everyone can read that. It makes total sense. But if we delete one nucleotide, so one letter, out of the entire sentence, the cat tet her at, it changes the entire frame, and so now it makes no sense. Everything down from where that frame shift mutation occurred, where that one deletion occurred, has changed. And so that oftentimes will cause stop codes to be produced, or the protein becomes non-functional. And here we have an insertion. The cat at eth erat makes no sense again. And again, you could produce that stop code. Another spontaneous mutation occurs when you have a jumping gene. So a transposon is a jumping gene, and it can change positions within a DNA sequence. It can also move from one DNA strand to another DNA strand, and this can cause all kinds of problems with the functionality of proteins. So quickly, here you see a transposable element in this plasmid here. It jumps into this plasmid, and you see there's this gene X. Well, gene X, when it jumps, gets disrupted, and so it's no longer going to be a functional gene. It won't work anymore. In bacteria, you can't cut out the non-coding portions like you can in eukaryotes. Therefore, there'd be nothing you could do. So that gene would not function. Transposons were actually identified by Barbara McClintock in 1983, and what she saw and how she found them was by observing corn kernels that had different colors, variations in corn color were associated with transposable elements moving. And again, if you want to watch this video, you can. I'm not going to play it right now. You just have to look it up on Google. Just type in transposons, shifting segments of the genome, McGraw-Hill. So more likely, we're going to see induced mutations. Most mutations that occur are induced, especially those that we care about because we're trying to cause a mutation. Mutations can be caused by either chemicals or by radiation. And oh, hello, wow. Um, Chemical mutations can cause both base substitutions as well as frameshift mutations. So here we have an alkylating agent that adds an alkyl group to a nucleotide. And so here we have guanine that always pairs with cytosine, but when you add an alkyl group to the guanine, it becomes a methyl guanine. And now we compare with cytosine, but we also compare with thymine, and that causes a point mutation. Base analogs are different. They're not the typical nucleotides, but they resemble those nucleotides, and so they can move in place of a nucleotide. So here we have thymine, which always pairs with adenine. So adenine and thymine always pair together, but fibromous uracil looks very similar to the thymine, and it can pair with both thymine or cytosine, causing another point mutation. Intercalating agents cause breaks within the DNA sequence and allow for either deletions to occur, or you can cause insertions to occur. This causes frameshift mutations. My favorite one that I talk about is ethidium bromide. Ethidium bromide is an intercalating agent, and it also is radioactive and very likely a serious carcinogen. So when you add, what we tend to do is in our electrophoretic gel, we add ethidium bromide to the gel when we're making it, and then we put our DNA sequences into our little wells, and we allow the DNA to run down from the negative side to the positive side of the gel. And then when we look at it under UV radiation, because ethidium bromide is an intercalating agent, it'll get in between the nucleotide strands, and it'll cause the DNA to glow at the different spots. So it's really cool. But if you're ever working with it, you have to be very careful because, again, it is highly mutagenic and very likely a carcinogen. Then we have ultraviolet radiation. Ultraviolet radiation, or we have radiation. There are two types. We have ionizing and non-ionizing. Ultraviolet radiation is non-ionizing radiation. And when it attacks the DNA, it causes thymine dimers, where two thymines in the same strand of DNA will attach to each other, and so they can no longer attach to the adenines. This can be fixed, but if it doesn't get fixed, then the cell will no longer be able to replicate its DNA and the cell will die. X radiation causes breaks in the DNA, single and double-stranded breaks. I thought I had a picture, sorry. In the DNA and can cause one, frameshift mutations, but otherwise it can also just be lethal because the DNA breaks up completely. So we have these different mutations. Spontaneous mutations are almost always repaired, but even induced mutations can be repaired, especially those mutations that are not caused in a laboratory when we're not actually controlling the mutation. We have all organisms have mechanisms in place to repair their DNA. The main mechanism that we use is proofreading by DNA polymerase. DNA polymerase is about 99% accurate and can move along the DNA strand. And if a nucleotide that is not supposed to enter comes in and tries to pair with the wrong nucleotide, then it gets kicked out by the DNA polymerase because it's being proofread, and then the proper nucleotide will come in. But then some organisms, different bacteria specifically, one species that I always like to talk about is E. coli, our favorite, has both light and dark repair enzymes that causes the repair of a strand of DNA that was relatively newly synthesized that has some type of a mismatch in it. So light or dark repair enzymes can work in mismatch repair. Light repair enzymes are going to be activated by the sunlight. Dark repair enzymes are activated in the dark. But these enzymes, what they do is they'll move along the newly synthesized strand of DNA. And how do they know it's newly synthesized? Well, the parent strands, after DNA has been synthesized, after a little while, the DNA will become methylated. So we have these little methyl groups up here. And so this side has no methyl groups, which means it's the daughter strand that has just been synthesized. And so these mismatch repair enzymes will come along and they'll see that, okay, there's a mistake here. They'll cut the DNA before and after that mistake and cause that section to move out of the area. And then DNA polymerase will come along and produce the newly synthesized strand with the proper nucleotides, and then ligase will come in and seal everything together. Seriously? All right, I'm not going to worry about this. Again, you can watch these videos on your own. So a mutation changes the phenotype. A phenotype is the physical characteristics of an organism. If you have a mutation, it can cause your physical characteristics to change. So if you're able to utilize lactose, if you have a mutation, you may no longer be able to utilize lactose. That would be an example. A prototroph is an organism that does not need any special growth requirements, growth factors. It can grow, it can produce whatever growth factors it needs, and it grows well. We often produce organisms that cannot grow properly without special growth requirements. These organisms are called auxotrophs. So an auxotroph needs some certain nutrient. Oftentimes it might need an amino acid that the wild cell could produce on its own, but these cells can't. Once we, and so why do we produce these mutants? Because we want to be able to find the organisms and then run tests on them. So an example, we may produce a mutant that is unable to grow or is resistant to all different antibiotics, so that then we can grow it and try to use new chemicals to kill it off. That might be an example. If we do that, then we also might want to make sure that that organism can't grow without certain growth factors, because we don't want it growing out of control. So we make it auxotrophic where maybe it needs histidine. We have to add the histidine, or we have to add the tryptophan, whatever amino acid, and it'll only grow with the tryptophan or the histidine. So how do we find these mutants? Because think of the size of bacterial cells. They're super, super tiny. I mean, microscopic. A lot of times when you're looking at those cells, you're like, I don't know if it's a rod or if it's a coccus. It's so small. It's like a speck, and that's 1,000 times bigger than what the size actually is. So these things are super small. And when we're playing with them, when we're trying to make them resistant, or when we're trying to produce these auxotrophs, we're doing it in a solution where we don't actually see the cells. So we have to then be able to pick those cells that we care about out. There are two mechanisms to do this. Direct selection, where we are looking for an organism, the only organisms that can grow. In that case, we're going to select an organism that will be able to grow on a media that other organisms won't. So here's an example, a media with media containing streptomycin antibiotic and a media without streptomycin antibiotic. If we're looking for an antibiotic-resistant mutant, then if we expose the mutant to the antibiotic that it's supposedly resistant to, only those cells should grow. Any cells that don't grow, or that don't grow, don't have that resistance factor, so we directly selected for it. Indirect selection uses auxotrophic bacteria, or is looking for auxotrophic bacteria from their prototroph parent cells. Auxotrophs do not have the ability to grow without certain chemicals. So we use a process called replica plating. We start with a colony or a series of colonies on nutrient agar, and we know that some of these colonies may have the ability or may not be able to grow it without all of the growth factors added. Once we have all those colonies, we mark the front of the dish. We then take a sterile velvet press it into the agar so we get a picture of all the colonies. Then we take a nutrient agar, plain, sterile, and a glucose salt agar that does not contain all the nutrients. So this one would be excluding the histidine or the tryptophan, whatever it is we're looking for, cells that cannot grow without histidine or tryptophan. We press it into both of these plates. We then wait for them to grow, so we know the front of the plates here and here. And then we look here, we have all the colonies that grew there, but on this side, there's a space where there was a colony, but there's no colony here. So that's the area that there is no cell growing, so we can come over here and find that cell. There's your auxotrophic mutant. It's a little different, but it does work. So now we know how to find these cells. Another thing we can do with auxotrophs is we can use them to see if certain chemicals may be, well, for one, mutagenic, and for two, if they're mutagenic, could they be potentially carcinogenic? So anytime we're putting new chemicals out on the market, we want to make sure that they're not mutagens. So you wouldn't want to use a mutagen on your kitchen counter. That would be crazy. So we take two plates, a control and then a test plate. We use bacteria that are auxotrophic. So in this case, we have histidine-dependent bacteria. We add the bacteria to the agar plate, and this agar plate lacks histidine. And so here we have glucose salt agar that lacks histidine. We use the exact same agar, add the exact same bacteria to both. We then, in the control, add a liquid that does not contain any mutagen, so this might be just water. And then over here we add a liquid containing the mutagen. So then what we're going to do is we're going to look at how many colonies form. So because this is a medium lacking histidine, and these are histidine-dependent bacteria, no colonies should form. But anytime you have cell growth, there's always the potential for some mutation. And so you have to look at how many colonies grew, and in this case, 2 colonies grew from all the billions of cells you put on there. Over here, if you only have a couple, 2 or 4 colonies that form, then you know that that's not very different from the natural mutation rate. So you don't have to worry. This chemical is likely, it's not a mutagenic, and so it's probably not carcinogenic. But if, on the other hand, you have tons of colonies formed, you had some mutagen caused of mutations in those auxotrophs, and now they're able to grow really well. That means that the chemical was a mutagen, and the more colonies that grow, the more likely it is a carcinogen as well. All right, I'm going to stop here, and I'll start on talking about transfer of genes via bacterial cells. Okay.